Okay, good evening, everybody. So, Nisha or anybody else, can you please guide me how to do that, the attended thing? So, I should click on the three dots at first.
Yes, sir. Then, sir, meeting info or any meeting option is there.
It's called Manage Permissions. So, I click on that.
Sir, meeting info.
Let me see. Meeting details. So, I was actually clicking on the people thing. So, you say I was asking, I think, the three dots below, besides the hands sign, right?
Yes, sir. Yes, sir. Yes,
Okay, it
sir.
says meeting details, meeting notes, and meeting settings, device settings. So I'm clicking on meeting details.
Yes, sir.
Meeting details, meeting options, yes. And then there are a lot of
Then, sir,
options. Yes.
then there is option, sir, at the end, sir. See,
Allow
who can
reactions.
who
Allow
can present,
who can
the
present. Or it was everyone. I should say that only me.
only me, only me.
Okay.
Then just save it.
Save it. Okay. Thank you. I did that. Thank you. Okay. So I'm going to share my screen and we'll start then.
Sir,
Yes, follow.
all student not present, sir.
So,
10 student present, sir. Now,
yes, so I'm asking you, so what can I do if they are not present? Hopefully, some of will join.
okay,
Meanwhile,
sir. Start.
okay, good.
Okay.
So, can you see my screen and the
Yes, sir.
PowerPoint?
Yes,
And
sir.
now confirm whether you can see my PowerPoint or not. My PowerPoint is loading. I hope you can see the PowerPoint
Yes,
as
sir.
well. Oh,
Yes, sir.
good.
Yes, sir.
Yes, sir.
Very good. So, I'm going into the full screen mode. Okay, so in the last class, basically we talked about the innate immunity thing, and a little bit at the end, I talked about the adaptive immunity. So I will finish the adaptive immunity part a little bit, and then I will move into other topic. So I hope you more or less now understand the difference between innate and adaptive immunity. In case of innate immunity, my slide is not moving, right? Is it moving?
No, sir.
No.
No, sir.
All right. No, and now it's moving, I suppose. Okay, so we have seen that in case of innate immunity, as I mentioned, that it's not specific, it's a very generalized type of protection. There's no specificity and it depends upon some pattern recognition thing. Of course, very important though, and it has some other kinds of cells that are involved like macrophage and neutrophils, etc. And it doesn't matter at all that whether you have been challenged with a particular organism or not, where you are born, whether you're Indian or American or Australian, whatever, it doesn't matter. As a human being, all of us have the same type of more or less innate immunity most of the time. But adaptive immunity differs from species to species, from different strains of the species or different population of the species, and most importantly, they are capable of recognizing and selectively eliminating. So that is the term we will remember, that they are selectively eliminating specific microorganisms. So if you have the adaptive immunity against E. coli, then you are protected against E. coli, but you are not protected against, say, another bacterium called Salmonella. That means if you compare with the security person, your security person is very much trained that if there is a person coming to sell something, say maybe sell biscuits, then you have to stop them. No vendor who is trying to sell biscuit should not enter into the complex. But if somebody is selling fruits, apples or oranges, then that security person will not bother about that. That person can enter easily. Okay, so if you want the fruit vendor also to not to allow to enter, then you have to train the adaptive immunity different way. Okay, no, this person also should not be allowed, something like that. So it's very specific about his functions. Okay, so they are not same for the all members as I mentioned, and adaptive immunity does not come into play until there's a challenge. And most important thing is that it has a memory. High degree of specificity, the remarkable property of memory. But I would like to say here that do not confuse with the memory in what we understand in case of human beings. Amader যেমন memory, as we have a memory segment in our brain, we can memorize something. There is a short-term memory, long-term memory, etc. there. And sometimes we amazed by seeing the capacity of our memory. Sometimes we can remember something that happened very long back, and that was not most probably a very significant thing, but somehow I remember. And sometimes we do not remember even whatever has been told yesterday. So that's a totally different thing. And that memory and this memory, memory has no relation at all. But we love to call it a memory because the way it functions, it looks like they have a memory. Okay. So 4 different characteristic attributes of adaptive immunity. I mentioned about these things before, because if you go into your previous slides, you will see that I mentioned about these characteristic attributes about the lymphocytes. So the same thing also here. So whatever the characteristic attributes of adaptive immunity is actually the characteristic attributes of lymphocyte B and T. No other cells are contributing to get these attributes. What are the attributes? Of course, the first thing I just mentioned now, that immunologic memory. Okay. Then we have a self and non-self recognition. I think we spent quite a bit time about understanding that they can distinguish between our own protein and a microorganism protein, a foreign protein. Okay, not only necessarily that have to be microorganism. Our protein and it's not our protein. Self, non-selves. Okay, so this thing. Number 3, you can say, if you go from the right side, diversity. The huge number of antibody molecules, possibility of antibody molecules are there. That I've also mentioned before, that every B cell that is coming out from the bone marrow is having a membrane-bound antibody molecule attached to it. Hope you remember, attached to it. And these membrane-bound antibodies are destined to are specific for a particular microorganism, a particular microorganism protein. But this particular microorganism, there's a chance that maybe doesn't exist in the universe. Okay, so that's why this B cell will be useless for your entire life, for our entire life, but it is there. If something comes up, it will bound to that microorganism or that protein. So this way, huge number of diversity of antibody molecules is found. And of course, the specificity. So this guy is specific to this. So unless this thing comes, he'll be useless, will not do anything. But another B cell, similarly, another antibody molecules, a membrane molecule, will never touch this antigen. Will never be able to bind to this. Okay. So you can think about that. If it is for E. coli, it is binding to the E. coli. And if it is specific to Salmonella, it is specific to Salmonella. It will never touch this antigen or this microorganism. It's so specific. So 4 different attributes: antigenic specificity, diversity, memory, and self, non-self recognition that you have to remember. Now, adaptive immunity and innate immunity, as we so far discussed, it seems that they're far away from each other. Their total logic of working, the function is different from each other. But they're not independent to each other. They work in a very cooperative manner with each other to give us a robust protection against different types of microorganism-derived diseases. Say, for example, the phagocytic cell. We have seen before the phagocytic cell like macrophage, that phagocyting different microorganisms. So they are a crucial part of mostly the innate immunity. Okay, so they do not have any specificity, they do not have any memory, they're also not a diverse type, so they only can sense the presence of a microorganism and goes at that place and engulf those microorganisms and destroys them. So they're very much a part of an innate immunity thing. But these phagocytic cells, which are crucial to the innate immunity thing, also are involved in the activating specific immune response. If you go into my first slide or something like that, when first maybe as I've shared with you, the person who proposed or who advocated that cells are also involved in immunity. who advocated about cellular immunity besides the humoral immunity. Then he commented, the person's name is Eli Metchnikoff. Eli Metchnikoff commented about the thing that whenever the macrophages, the phagocytic cells, are getting activated, they're also showing a robust specific adaptive immunity as well. So there's a relation. Phagocytic cell, part of the immunity, but when they're getting involved, getting activated, they're also activating our adaptive immunity in turn. Macrophage or phagocytic cell, part of innate immunity getting activated or involved, and then in turn activating the adaptive immunity. So adaptive immunity is partially depending on the innate immunity. Okay. On the other hand, the question is whether the innate immunity depending on adaptive immunity or not. Yes, there is also some example of that. Different types of chemical factors, soluble factors that are being produced by specific immunity cells, which is called adaptive immunity or acquired immunity. Those chemicals has been found to show that they are also activating the phagocytic cell. So it's working in a loop-like thing. Look for a circle type of thing. So I'm trying to say, so this is our macrophage. So macrophage has engulfed some bacteria over here, okay, part of the innate immunity thing. And then this macrophage is actually seeing that they are showing this microorganism to an adaptive immune cell part that is called T cell. So T cell is recognizing this microorganism. Yes, that is indeed a foreign one. You have done a good job, and it's releasing certain chemicals, and those chemicals are reaching to the macrophage, and this macrophage is becoming more excited and more activated and doing this job even more efficiently. So it's working in a circle. Innate immunity component helping or activating the adaptive immunity, and adaptive immunity component in turn also helping the innate immunity part. So both these type of immunities so kind of interdependent, but not entirely. They can also work in a little bit of independent as well. Okay, so now an effective immune response involves two major group of cells. Just what I drew, I will show the same thing. T lymphocyte and antigen presenting cell. So maybe you are hearing about the antigen presenting cell or APC for the first time, and you may wonder where this antigen presenting cell is coming from. Let's see. So two group of cells we require. One is T lymphocyte, another is antigen presenting cell. So actually what happens if you look into this picture, as I said, T cell cannot bind to any free antigen. Somebody needs to present that antigen to the T cell. Whereas the B cell can bind to a free antigen. So let's imagine this is our B cell. So this is our B cell. B cell must have a membrane-bound antibody through which it is binding to the free antigen. A microorganism may be just entered into the body and then processing the microorganism into small protein part. And then it's presenting that protein with the help of MHC molecule. I've talked about the MHC a little bit before, major histocompatibility complex. So to whom? To T cell. And what we have in the T cell? We have T cell receptor or TCR. So T cell will bind to this. So who is presenting? B cell is presenting. And what you have seen here is antigen presenting cell. So actually B cell is one type of antigen presenting cell. B cell cannot do much unless it's present the antigen to the maybe a superior position person, that is T cell. So it's waiting for the approval from the T cell. T cell will bind. T cell will say, yes, it's indeed a foreign microorganism. You can take necessary action to neutralize it. So present that there is antigen presenting B cell. But just what I have drew in the white skin a little 5 minutes back, the macrophage are also doing the same thing. Macrophage is also engulfing a bacteria and then presenting it to the T cell. So macrophage is also one type of antigen presenting cell. So there are certain cells also that are called dendritic cell. So certain type of cells are there which is known as antigen presenting cell, such as B cell, such as macrophage, such as dendritic cell as well. Okay, so that's why I was saying that an effective immune response always involves two major group of cells at least, T lymphocytes and antigen presenting cells, which is acronym is APC. Okay. So if you look into or focus into the antigen presenting cells, you can understand that antigen presenting cells are very, very important. If they do not present something, if something happens of this place impaired, then even though you have a very healthy, good T cell, nothing will happen. This reaction will not take place. So that's why antigen presenting cell is very crucial. So antigen presenting cell first internalize antigen either by phagocytosis or endocytosis and display a part of that antigen on their membrane bound to the class 2 MSC molecule. So we have to remember here that whatever you're talking about the MSC molecule over here is a class 2 group. Class 1 is totally different. Maybe I mentioned or maybe I didn't. I will do that later. So class 2, two types of MSC molecules are there, class 1 and class 2. So antigen presenting cell first internalizing the antigen by phagocytosis, then displaying a part of that antigen on their membrane bound to the class 2 MSC molecule. Then who is recognizing? If you are talking about the class 2, then immediately you cannot actually just say T cell. You have to mention a subpopulation of the T cell. T cell has two different populations. One is known as TH and another is known as TC. TH means T helper and TC means T cytotoxic. The role of TH and TC is entirely different. Okay, so the reason the name is pretty clear here. Helping, that this is helping. Okay, and cytotoxic, cyto means cell and toxic, you know that. That means that those cells are going to kill the cell. Okay, so here you are not going to kill the cell, going to help. So here, this cell actually T helper, and this T helper is going to help this cell, not to going to kill this cell. So this is the role of T helper, not cytotoxic. Fine. So whenever there is a class 2 MSC molecule is involved, there must be T helper cell and not TC. So T helper cell recognize and interacts with the antigen class 2 MSC molecule complex on the membrane of the antigen presenting cell. Antigen class 2 MSC molecule complex. So now you imagine this is this is our class 2 MSC molecule and this is our antigen. So these two is forming a complex. T helper cell is going to recognize this class 2 MSC molecule plus antigen complex. If you remove this class 2 MSC molecule and replace with class 1, T helper cell has no interest in that. Okay, for that we have T cytotoxic cell. That's a totally different ball game. So here, that's why I'm saying here that T helper cell recognizes with and interact with antigen class 1, class 2 MSC molecule complex on the membrane of the antigen presenting cell. Fine. Now, whatever I just drew in a white shape, the same thing, the macrophage engulfing a bacteria, processing the bacteria in a small portion or its protein, and then it displaying. What are the green things? Green are all NHC molecule. That means B for macrophage, B for B cell, they already have this MSC molecule inside. So the binding to the MSC molecule, and now you can see that displaying it. So now once again we are getting MSC class 2 antigen complex. And who is reacting to that? Who is binding to that? Helper T cell. Through which? Through what? Through T cell receptor. Okay. So here this is also a APC. So it's a macrophage. So macrophage, B cell, both can work as a successful APC. So what are the characteristics of successful APC? What you can say that this cell is APC. So B lymphocyte, macrophage, and also dendritic cells more or less are APC. First, they must express class 2 MHC molecule. So unless you have a class 2 molecule in your membrane, you cannot present that to the T helper cell. So you have to have a class 2 MSC molecule. That is the one criteria for being an antigen presenting cell. Secondly, that we will not go into detail, but if we take again the old example, what I like to say, maybe it's not much meaningful, that if you remember the T cell I said the Maharaja, so Maharaja has been served the food, somebody needs to serve, who is serving? B cell or macrophage as antigen presenting. Okay, so not only you have to serve the food, you have to use a specific utensil, maybe a golden bowl. What is the golden bowl? The MSC molecule. That's fine. And then also your Maharaja is actually sleeping, so you have to poke him. Okay, Maharaja, wake up and have your lunch. That waking up signal is called co-stimulatory signal. So another signal you have to use, that is called co-stimulatory signal for the activation of T helper cell. So this is our APC, either B cell or macrophage or dendritic cell. So here you can see this red one is our peptide, means the antigen or microorganism. This is the MSC class 2. This is a TCR, so they're reacting, very good. But still, T cell will not be activated. You have to provide another signal, this violet color thing, which is known as co-stimulatory signal. So unless this co-stimulatory thing works, T cell will not be activated, even though this will be working fine. So two different criteria for being the APC. So we understood the reason for having the APC. Who are APC? Macrophage, B cell and dendritic cell. Then are looking for the two properties. One property is that they have to express MHC molecule on their membrane, major histocompatibility molecule class 2. And number 2, they are also have to able to deliver co-stimulatory signal for T cell, necessary T helper cell activation. So this type of specific mention of T helper or class 2 is necessary. Otherwise, it could be very confusing, because if you do not say class 2 and MSC, then people might think that you do not know about the two different types of class 2 class of MSC molecule, or you do not know about the two different subpopulation of T helper cell. So that is the APC thing. So if you look into the whole scheme once again, so you will be able to understand. We have used this picture before as well. So here our B cell, you also can think about as a APC. B cell with its membrane-bound antibody is binding to a part of the bacterium, microorganism, fantastic. Then what is doing is actually processing and making a small fragment. And one of those fragment is being presented with the help of class 2 MSC molecule. To whom? To the helper T cell. Okay, helper T cell is binding with this T cell receptor or TCR. At the same time, they're also producing a co-stimulatory signal that is not shown over here, but this CD4 is ready for receiving the signal. So now T cell receptor is getting activated, successfully activated. Recognize a foreign microorganism, presented to it through MSC class 2, and also got the access stimulatory signal. Now T cell what is doing? What is the meaning of activation? T cell what will do? It will release chemical substances. Look at it. You can take all these chemical substances, you can put them in a single class, name as cytokines. You must have heard about these cytokines in this COVID discussion somewhere, cytokine storm, etc. So these cytokines are a class of molecules. We do not have the scope to discuss about those chemicals very much. So these cytokines will release. Then those cytokines, that is not shown over very clearly in this picture, these cytokines will go again and bind to the B cell receptor. Or if you imagine that this is a macrophage is doing the work as APC, this cytokine will bind to the macrophage. Now, we're not talking about the macrophage anymore. If you think about the B cell, after receiving the chemical, so you see, these two cells are actually talking with each other. They're communicating with each other, but they're not human being or any other thing. They cannot talk. They do not have any language. They only understand one language, that is chemical language. So they're presenting something, they're recognizing, they're agreeing, yes, It's okay. It's really a foreign microorganism. We need to neutralize that. So you're releasing some chemical signal, and that signal is reaching to this B cell. Then what will happen? Then the B cell will differentiate into two types of cell. One is known as plasma cell, also as effector cell. We know that these plasma cell will produce soluble antibody. So these antibodies are not anymore bound to the membrane. Okay, soluble antibody. But this antibody is actually the exact replica of the same antibody. Why that is required? Because here we are talking about specific immunity. So if you want to bound, if you want to neutralize this bacteria present anywhere in the body, anywhere in the blood or lymph, then you have to require the same exact antibody. So now this antibody will started that journey. Antibodies that started that journey. And anywhere in the system, they will find this microorganism present. They'll be able to successfully bind to that, okay, and try to neutralize. That is the role of plasma cell. But on the other hand, another class of cell got differentiated in the memory cell. So they are not at all releasing any soluble antibody. They are only being there for a long time with the same membrane bound antibody. So one particular clone of cell, one particular group of cell will be there for a very long time. Okay, so they have a long time lifespan, whereas the plasma cell have a very short lifespan. So they will be there. So in any time in the future, if the same microorganism attacks, invades our body, then it will be very easier to distinguish them. You have distinguished them, and also you have a full battalion ready to combat. Here you only got one or two or some 100 or something like that. They are trying to neutralize, internalize them, trying to process that, trying to present that to T cell, and then the T cell is doing, but number of cells are very low. Okay, so you have to repeat the same process, but now you have a whole battalion of cells. B cells is ready for this microorganism. So whenever will come, all these battalions will be readily able to bind to them and will send a signal to the T cell that naughty bacterium entered again, give us the signal. T cell will provide the signal, and within a very short period of time, huge number of antibody molecule will be produced, and the antigen or microorganism will be reduced or neutralized in a quick time. So that is called secondary immune response. So there is a graph, maybe I mentioned, I do not know. So if you think in case of the, if you think that this is our, in x-axis, we have the amount of antibody produced, and this is our time. Okay. So number of antibody produced with time will be a very less in the initial time, first time when you are encountering. But say after 6 months, the same bacteria encounter, so gradually your antibody titer, antibody concentration is decreasing. But the second time, when it will be like this, very sharp peak within a short period of time. The reason is that we have a full battalion of cell ready for that. So that is the basic thing that is our vaccine concept relies on. Okay, so you will not be that much suffer from that much, you will not develop that much of symptom. Okay, whenever there will be a bacterium or virus will come, you already have the antibody bound memory cells, so they will be able to pick up, they'll be able to recognize the organism very quickly, and they'll be able to take necessary action. Okay, so antibody functions, we can think about what is antibody doing. So antibody, maybe I mentioned once before, that antibody is not anything which is killing the microorganism or destroying the microorganism. Antibody is only making a facilitate the destruction. Functions as an effector of the humoral response by binding to the antigen and neutralizing it or facilitating its elimination. Most of the time, it's only facilitate its elimination. That means helping its elimination, not eliminating it actually. So these green things are all our antigen or microorganism, and the violet ones are antibody. So they are binding this. So when you can see the antibody here are coated with antibody. So when it is coated with antibody, it helps the other organ, other cells to eliminate it much easier way. Say, for example, phagocytosis, macrophage. So macrophage will ultimately engulf them, will eat them, will destroy them. But it is becomes very difficult for the macrophage to individually find out and engulf one bacteria or virus, etc. So now what we have seen, that antibody molecules are coating the antigen and make a group of microorganisms as a blob. So it will be easier for a phagocytic cells such as macrophages to come and to engulf the whole group easily, rather than finding out each one antigen or microorganism. So antibodies helping those phagocytic cell to kill or eliminate, because macrophage has lot of different things to kill the microorganism. Antibody do not have any such thing. Antibody only can facilitate this thing. So they're cross-linking antigen. Another thing is that sometimes what antibody does, that when antibody is coating this antigen, the bacteria cannot move much. They lose their mobility. They cannot move from one place to another. So that's why they also cannot replicate. And that's also one type of neutralization. But another thing is that if the microorganism is releasing any toxic material, so all the time microorganisms do not work in a similar fashion, but some of the microorganisms, they release toxin. Say, for example, the Vibrio cholerae, which is responsible for the disease cholera, releases cholera toxin. For diphtheria, Corynebacterium diphtheriae, they release diphtheria toxin. Tetanus, the bacterium releases tetanus toxin. So antibody can also neutralize those toxin products, or sometimes virus particle, by coating them, which prevents them from binding to the host cell. Okay. So this is the whole scheme about the innate and adaptive immunity that I was thought about talking, and to finish the earlier part. So I will stop here for the first part, and it's the time for you to provide attendance, and then I will come back. So, okay, let me find. So it's 7/7. Fine. Today is 8th September. You check and let me know whether it's the attendance form. I have posted in the chat room, chat box. Did I post the right form?
Yes, sir.
Okay, okay. So do the needful. I'll be back in 5 minutes time.
Hello, sir.
Sir, the form is not opening.
Yes, sir. This is
Okay, I have corrected it. There's a problem with the timing, so I'm pasting it again. Now it should work. Is it working now?
Yes, sir.
Okay, good. Okay, I'm back. So hopefully you can see the PowerPoint and can hear me. So in the next part, we'll be talking about antigen and immunogen. So just confirm me, is everything okay? Can hear and see this PowerPoint?
Yes, sir.
Oh, good.
Yes, sir.
Oh, good. Thank you. So we are talking about this antigen several times, and more or less we're using the antigen word as a synonym to the microorganism. That is not entirely true. And then another term is also immunogen. So we'll try to understand a little bit about the differences. Is there any differences or not? So the question, of course, comes classically, what is antigen? So you can think that substances that can be recognized by the B cell receptor or by the T cell receptor, of course, when complex with MHC molecules are called antigen. So if anything that could be a microorganism, may not be a microorganism, a simple protein maybe you have inserted in animal system, whatever. If that is recognized by a B cell receptor, that means B cell membrane bound antibody, or the T cell when complexed with the MHC molecule is called antigen. Okay. So unless one of these two options are fulfilled, we cannot call them as antigen. Most of the time, the microorganisms are considered as an antigen because they are being recognized by the B cell receptor and then presented to the T cell. They'll be recognized by the T cell receptor as well. Okay. So then, what is immunogen? Immunogen is substances or antigen, you can think, which have the ability to induce a humoral or cell-mediated immune response. So now that becomes a bit confusing. So here, you can imagine that maybe this one thing that has been recognized by the B cell receptor, when presented to the T cell also can recognize it, but they are not getting activated. They are not taking any action. They are just idle, sitting idle over there. Okay, maybe the B cell is not processing the antigen, or maybe it's processing when T cell is not getting activated. So they are not activating our immune response. They're not stimulating our immune response. If they are not doing so, then you cannot call them immunogen. So only those antigens or those substances which have the ability to induce or stimulate whatever branch of the immunity, cell mediated or humoral, is known as immunogen. So here antigen is very easy. Just by fulfilling one of these two options, they could be called classically as an antigen. Either can be recognized by the B cell receptor or by the T cell receptor when complexed with MHC molecule is antigen. But here the definition or question is much more crucial. The question is, are they inducing our immune response? Are they stimulating our immune response? If yes, then they are immunogen. If no, then they are not immunogen. Then the question comes, is there anything like, so you can simply say one thing, that there be a lot of things which are actually antigen but not immunogen. That means they are getting recognized by these two, but they are not capable enough to induce our immune system. So all the antigens are not necessarily immunogen, but all the immunogen is of course antigen, because that is the thing we started with the word. Okay, all the antigens are not immunogen, but all the immunogens are necessarily antigen. So immunogenicity and antigenicity, just we're using the term as because of the capability. You can try to understand that what is the immunogenicity. Immunogenicity is the ability to induce an immune response. Immunogenicity is the ability to induce an immune response, be it humoral or be it cell mediated. Say for example, here this is our immune cell, B lymphocyte, and this is our antigen. Okay, so when they are reacting, you know already what are the steps is in between. Finally, what we're getting? We're getting plasma cells and memory B cells, and we know that these plasma cells are going to produce soluble antibody molecules, and these soluble antibody molecules in turn is going to bind to this antigen wherever they will find them. Okay, so they are inducing, they're stimulating our immune response. Our immune response, our immune system is taking action against this antigen. So that capability or that ability is known as immunogenicity. But antigenicity, antigenicity is not like that. Antigenicity is only the ability to combine to or to react to specifically the final product of this immune response. So final antigenicity is the ability to react to the final product. What is the final product? Final product is antibodies. There are also cells of the receptor, forget about that. Antibodies. Okay, so you remember what I said, this plasma cell will produce antibody, and this antibody look for the specific same type of antigen everywhere into the system, and this antibody will bind to this antigen. So they are now, what is doing? They're binding to the final product. So they are antigenicity, showing the antigenicity. In this case, they have shown the immunogenicity. So antigenicity is only the ability to combine specifically with a final product. Okay, but they are not necessarily involved in stimulating our immune response. If they are involved in the stimulating our immune response or inducing our immune response, then we can call they have immunogenicity. So immunogenicity, antigenicity, different. Immunogen, antigen is a different. Okay, so that's why I was saying that all immunogens are antigens, but all antigens are not necessarily immunogens. So although all molecules that have the property of immunogenicity also have the property of antigenicity, but the reverse is not true. So this is a small story that you can try to read, or I can say just a funny story to make you understand about the immunogenicity or antigenicity. You may like it. So I like it. That's why I share it with the students. So say, for example, you are from the morning you are nagging and you are requesting your mother to prepare some homemade pizza, say for example, or you can replace with any other type of food. Okay, so you are encouraging, you are requesting your mother to prepare that. So in the evening time, your mother prepared a hot pizza fresh from the oven and kept on the table. Very good. And then your dad. So I'm just imagining here that your mom is working from home, or maybe a housemaker. So your father came back from office in the evening, and after washing his hands and etc., he just took a bite of the pizza and started eating it. Okay. Now you see, from the entire day, who was inspiring your mom? Who are inducing your mom to prepare the pizza? It's you. Okay. And what the role your dad playing here? Nothing. He didn't inspire at all. He just came from the office and just took a bite. So here the pizza is a final product. And who is consuming that? Your father. And you will also consume later. No problem with that. So your father here is antigen, who is binding to the final product. Okay. But he played no role in inducing the B cell or inducing the other T cell maybe to produce that pizza. Who inspired you? So you are immunogen. You also will bind. You also will consume the pizza. So you are immunogen who has the ability or who played the role to induce the system. Okay. And then also to the bind. But your dad is only played the role to bind to the final product, that is consumption of the pizza. So that's a funny story, maybe not very good, but it might help you to understand the difference between immunogen and antigen. Okay, so you may ask, is there anything which is really antigenic but not immunogenic? Yes, there are. So certain molecules, very small molecules, which is known as hapten. Okay, hapten are actually antigenic, but they're incapable of inducing any specific immune response. Okay. So that is hapten is antigenic. That means they will bind to the final product. If you can produce some antibody against hapten, then hapten will bind to those. So hapten is showing antigenic. But as an individual or in a group, hapten do not have the ability to induce our immune system. So they are not immunogenic. Actually, what happens? That's why you have to use some kind of protein carrier, okay, to use the to produce the antibody. So I hope you understand what I'm saying. So if hapten do not have the ability to induce our immune system, then how you will get an anti-hapten antibody? Okay, then how you can say that hapten will bind to those antibody? Because what you are doing, you are using some carrier protein. You are using a carrier protein and you are binding some hapten molecules to this carrier protein. So now they become immunogenic. Now our immune response system will be able to recognize it and will be able to produce anti-hapten antibody molecules, and those antibody will bind to the hapten. So hapten is antigenic but not immunogenic. This happens for all different type of drugs, etc. Say for example, the steroid molecules that the athletes usually takes, sometimes takes for better performance in athletics, which is called drug or dope, we can say. So for those things to figure out whether the person who just completed a 100-meter race, maybe below 10 seconds, whether it's taking any type of those forbidden drugs or medicines or not, then people usually take out blood and urine and they try to figure out whether there is any steroids or not. So those steroids are haptens, okay? Those steroids cannot produce or anything in our immune system, but in the forensic laboratory, we already have antibodies against those haptens. So if you take some urine sample and if you use the antibody, and if something comes up, then it will be very clear, easily saying that yes, that particular person was taking some not allowed substances. So these are the things. So antigen, immunogen, antigenicity, and immunogenicity. Now, then what are the factors that influencing the immunogenicity? We understand that yes, all antigen do not have immunogenicity. Then what are the factors? There are several different factors. Maybe we can categorize into 5 different parts. So one introductory line here is that immunogenicity is not the intrinsic property. Intrinsic property means inherent property, but rather depends upon number of properties of a particular biological system that the antigen encounters. So it is not an inherent or intrinsic property of an antigen to be immunogenic. That means an antigen, whether it will be able to stimulate our immune system or not, is not at all an inherent property. But that property actually depends upon some other type of property of a particular biological system, maybe that the antigen is going to encounter. Okay, fine. So let's see what are those properties. We can talk about 5 different properties. Maybe we'll talk about 4 different today. We'll leave the 5th one for the next class. 5 different, what property are you looking at? Once again, we have understood the differences between antigen and immunogen. We have understood the difference between antigenicity and immunogenicity. And we're stating that immunogenicity is not an inherent property of an antigen. Okay, that property and the immunogenicity depends upon number of properties of factors. And what are those factors? There are 5 different factors largely. Okay, those factors known as foreignness. First one is foreignness. Then the second one is called molecular size. Third is chemical composition. Fourth is antigen processing and presentation. And the 5th one is biological system. Okay, foreignness, molecular size, chemical composition, antigen processing, presentation, and biological system. These are the 5 major properties upon which immunogenicity of an antigen depends. Let's think about the foreignness. So if you can say in Bengali, you can say to me bideshi, kotokhani bideshi, something like that. How foreign you are. So in order to elicit an immune response, a molecule must be recognized as a non-self. We know that. We must be talking about adaptive immunity here. We know that our immune system is very specific. They have the attribute or characteristic to recognize between self and non-self. protein mostly. So they have the property. Now, let's take this example. I hope you know that proteins are made up of amino acids. We have 20 different amino acids in our system, and all our proteins are in combination of those 20 amino acids and a different length. So we are considering here that for one protein that is actually our one own protein, say a muscle protein, that is has a amino acid sequence A, B, C, D, and E. So 5 amino acids A, B, C, and D sequentially they are placed one after another and form this protein. So far from this discussion, you understand that our muscle protein A, B, C, D, E will be never recognized by our immune system as an antigen, okay? Because it's our own protein. So this cell protein has no immunogenicity. They do not have the capability to induce our immune response. And that is good, because otherwise we'll be suffering from autoimmune disease. Okay, fine. Now one microorganism entered in your body, and that microorganism has a protein which has a combination of A, B, C, G, and E. So only thing happened, the D amino acid has been replaced with G. Okay, so 1/5 change has been happened. So we can say that this protein coming from a microorganism, say X, is kind of a 1/5 foreign in comparison to others. So it will be difficult for our immune system to recognize sometimes this subtle change of one amino acid. Okay, so it may fail to understand that actually this is a foreign protein because the differences are very low. So definitely this is a foreign, but not very much foreign. So now another organism Y came, and it has an amino acid combination of a protein A, B, S, G, E. So here you can see 2 amino acids got replaced with S and G instead of C and D. So it is a little bit more foreign. So there is a chance that our immune system will be able to recognize that this is a foreign protein. Then another amino acid came as Z. It has a combination of A, X, S, G, E. Say for example, so out of the 5, 3 amino acids are changed. So we can call this is the most foreign protein, this is a more foreign protein, and this is just a foreign protein with respect to our cell protein. Now what happens in terms of our immune system, B cell or T cell, they of course will not touch this protein because this is our cell protein. They may or may not touch this protein as well. They could be confused. They may not figure out that there's a subtle change. They also could be a little bit less aggressive about this protein because they have been trained in such a way that do not touch anything cell protein. So they're very cautious. But for this guy, they're very sure that this is definitely a foreign protein. So they will jump on it. Okay, so you can see this foreign protein, first one, will show very little immunogenicity. They are not stimulating our immune system that much. This is a little bit of immunogenic, and this is a mostly immunogenic. So degree of foreignness is determining whether an antigen will be immunogenic or not. So when an antigen is introduced into an organism, the degree of its immunogenicity depends upon the degree of its foreignness, how foreign it is. So you'll be interested to know that maybe I mentioned in the very first class or so that in the fun of studying immunology is that in the both end we have some live organism. Okay, so in one hand we, the vertebrate multicellular organism, human being, and what we're dealing with bacteria, virus, protozoa, fungus, all are living. So they are changing, they're evolving as well. So it's not a biochemical reaction sometimes like that you are taking some copper sulfate and putting some sulfuric acid to that, etc. It's not like that. So both way, it's a very alive thing. So that's why what happens, both the group is evolving. So they're taking some, both of them changes the strategy to beat one another because it's a matter of survival. So microorganism also wants to survive. They want to go into our body. We also want to survive. That's right. So this is also kind of a strategy. So if somehow one microorganism can evolve in such a way that that this jade microorganism was very vulnerable. They know that I have the AXSG combination, and if I enter into the human body, I'll be easily caught because I have a very big difference with the actual protein. So gradually they evolved, and I'm not going into the evolutionary thing, how they evolved, etc. They try to reach to this place, at least to this. So here they have replaced their XS with a BC. So they only have one difficulty over here. So now they have a fair chance to survive. They can cheat our immune system. They can escape recognition by our immune system. So that type of strategies always microorganisms takes to escape the defense system. Okay, so there's some examples here. So maybe whoever is about the biology thing, that we in the laboratory use bovine serum albumin. Bovine serum albumin means bovine means a cow, sheep, a buffalo, etc. So you can take some blood out of those animals and you can isolate as albumin protein. Okay, so that is called BSA. So now you took this bovine serum albumin protein and you inject it into a cow. Then what will happen? Will the cow immune system will react to that? No, they will not react because this is coming from the cow. So a protein coming out of a cow and then entered into another cow. So this cow immune system will not recognize this BSA as foreign. Okay, so no immune response will take place. Nothing will happen. But if you take this BSA and then put it into a rabbit animal, then the rabbit immune system will definitely recognize this as a foreign one. It's okay. So it depends. So generally, you can say that phylogenetic distance means what type of animals we're talking about. So there's a small example. So you can see this called phylogenetic tree. We are here close to the chimpanzee group. Okay. So rabbit is also pretty close to us, but cow is much far below in the phylogenetic tree. That's why cow protein will be recognized as a foreign protein in the rabbit system. Cow protein might be recognized as a foreign protein in pig and dog. Similarly, a chimpanzee protein might be recognized as a foreign protein in cow. So this depends upon this phylogenetic tree or position. Fine. So the first we get down, so we thought about 5 different properties upon which immunogenicity depends. We discussed about the foreignness. Then comes the molecular size. Okay, so how big is your antigen? That will define whether it will be immunogenic or not. It is found that one antigen could be an immunogenic substances. It can induce our immune system if it is reaches to a critical mass of 5,000 to 10,000 Dalton. Anything smaller than that will not be a good immunogenic substances. You remember I said the small hapten. Haptens is very small. Steroid hormones are very small. Okay, so that's why if you take the steroid hormone injection, you will not generally will be your immune system will not reject it because your immune system will not be able to recognize it as a foreign one. Because it's very small size. You have to reach to critical size of 5,000 to 10,000 Dalton, but it's good to go for 1 lakh Dalton actually. So correlation is there between the macromolecule size and its immunogenicity. Generally, any substance with a molecular mass less than 5,000 to 10,000 Dalton are poorly immunogenic. They will not be able to induce our immune system that much. Okay. So after foreignness and molecular size, chemical composition and heterogeneity. Okay. So in case of protein, we have said that 20 different amino acids are there and different proteins are forming with their combinations. Similarly, also in case of sugar molecule, if you already have studied, then it's good. That's glucose molecules can attach to each other and form a long chain, and that way they can form cellulose or starch, etc., or maltose, etc. But sometimes what happens, different types of sugar or protein molecules can come together and form a molecule. So if there is only one type of sugar or amino acid, then we are calling it homopolymer. Homo means same. So say for example here, you can imagine that all these amino acids are same. They call it valine. Valine is a type of amino acid, VAL. So all are valine. Okay. So this is a homopolymer. But here you can see different colors. That the one is valine, another is alanine, another maybe it's also valine. Then we have proline. Then once again valine. Then this is I called alanine. And this is isoleucine. Then this is leucine. Then valine again. And then this tryptophan. So several different amino acids are forming a protein. So this is called heteropolymer. Homopolymer, same type of sugar or amino acid. Heteropolymer, different type of sugar and amino acid. Now it was found that heteropolymers are always more immunogenic than the homopolymers. As a simple rule, you can think whenever the molecule will be more complex, they'll be more immunogenic. So copolymer composed of different amino acids of sugar are usually more immunogenic, more chemically complex and more immunogenic. So we have seen so far, foreignness is critical. It will be more immunogenic. Immunogenicity will depend upon how foreign it is with respect to our cell protein. Number two, we have discussed about the molecular size. Our antigen must reach to a critical size of the molecule, such as 5,000 to 10,000 Dalton, ideally 1 lakh Dalton. Anything beyond that will not be immunogenic. We are now seeing that chemical composition is also plays an important role. Homopolymers are less immunogenic. Heteropolymers are more immunogenic. So chemical complexity is also a determining factor to figure out, to determine what will be the immunogenicity of an antigen. Fourth one, susceptibility to antigen processing and presentation. Let's try to understand that the development of both humoral and cell-mediated immune response requires interaction of T cells with antigen that has been processed and presented together with the MHC molecule. We just in the first class discussed a lot of things about this picture and we understood this. Fine. Now, large insoluble macromolecules are generally found to be more immunogenic. A large part, more or less, we have already covered under the molecular size thing. It also should be insoluble. It should not be soluble one. Large insoluble macromolecules are generally found to be more immunogenic in comparison to small soluble ones. Why? Because the larger molecules are more readily phagocytized and processed. So now try to correlate between these things. We have just discussed that macrophage or any type of phagocyting cell plays a role of antigen presenting cell. Fine. So it's very much required them to engulf different type of microorganisms. We have also said that it becomes difficult for a macrophage to engulf one particular bacterium or two particular bacteria. The jobs become easier for them if antibody molecule coats all the antigen and forms a big bubble type of thing for them. Okay. So similarly, with the same correlation, we can say that when the microorganism, macromolecules or the antigen is big, large, and also insoluble, then that makes the job easier for our macrophage to go and to engulf them, rather than if it is a small molecule and also soluble. So they cannot process it properly. If they cannot process it properly, they cannot be properly present it to T cell. If they cannot present it to the T cell, then nothing will happen. Whole the reaction will collapse. Okay, so nothing will take place. Although we have the microorganism or the macromolecule is still there, but they will not be processed. So no immune reactions take place. So they are lacking the immunogenicity or they're poorly immunogenic in comparison to the big one. Big ones are advantageous for us as our macrophage can see them very clearly, can engulf them clearly. So macromolecules that cannot be degraded and presented with MHC molecules are known as poorly immunogenic. Okay, so that is all we have for today. So basically what we learned in the second part is that what is antigen and what is immunogen, or rather what is the difference between antigen and immunogen. We have seen that anything that is being recognized by the B cell membrane bound receptor or T cell membrane bound receptor when complexed with the MSC molecules could be called antigen. On the other hand, if you have to qualify for an immunogen, you have to be capable enough to induce our immune system. So any antigen or any substance which can induce our immune system could be called an immunogen. So in terms of the adjective form, if we take it, so antigenicity means you are able to bound only to the final product, that is the antibody, then you are showing antigenicity. But you are capable enough to induce our cellular or humoral immune system, then you are antigenic, you have the antigenicity. And so you can see that every immunogen is actually an antigen, but all the antigen is not the immunogen. And we have a very classic example for that, that is called heptane molecule. Then we have seen that immunogenicity is not an intrinsic property or inherent property of an antigen. Actually, the immunogenicity depends upon 5 different types of properties, namely foreignness, molecular size, chemical complexity, susceptibility to antigen processing and presentation, and the biological factor. And we discuss about those things, how those are affecting the immunogenicity product, immunogenicity characteristic. We only need to discuss about the biological factor. There are 3-4 different components of the biological factor that will be discussed in the next class. Why biological factor is important? Because, you know, as I said, they are not attacking any iron or steel or plastic or something like that. They are dealing with biological things. They are invading human being or dog or pig or rabbit or cow, whatever. They also have the immune system. Okay. So biological factor is very much important, especially when you deal with that, when we work in the laboratory, the immunological thing. Okay. So that is all I have for today. And you can definitely ask if you have any questions or anything. Anything at all? Okay, then that is all for today. And maybe every week I say that I may not be able to take class, and I then figure out something to take the class. So what I will do, I will not take the class in the next week, more or less for sure, 90% sure. Okay, but I will give you a job through WhatsApp that you have to do an assignment. Okay, you have to write an assignment. I haven't decided about the topic or anything like that. That assignment carries 10 marks. So I will define the assignment, so you can write it in 1000 words or 1500 words, something like that. So you can write it in your hand and scan it and send it to me, or if you wish, you can type it in the computer in a Word format and send it to me. No issues, whatever you like. But one thing you have to be careful about. So I know whenever I say write something about B cell, you will go to the Google or you go to the ebooks what you have, and you will start copying words from those things. So that will not be acceptable. Be careful about that. I will do the plagiarism check from my end. I will check with the plagiarism software. So if I found anything that is you're getting match with any internet resources or book more than 20%, then that will not be acceptable and will be penalized. So be careful about that. I am not asking for anything that you have to write as an expert. I know that you are not an expert in immunology. It's just a GE class. So do not worry about those things. Okay. I want to see honest intention, honest effort from your part. You are definitely consult different internet resources. How many internet resources you will like, do that. But write in your own words whatever you understood after reading those parts, then write in your own words. Do not copy it verbatim, word by word, either in handwritten way or in a copy paste. That will not be acceptable. I will do the plagiarism check, and if anything goes more than 20% or so, that will not be acceptable. Am I clear?
Yes, sir.
Okay, good.
Yes, sir.
So I will send the assignment topic and you will get plenty of time to do that. There's nothing to worry about. Okay, so that's all for today.
Okay, sir. Thank you, sir.
Thank you. And maybe see you on the 19th, most probably, if I do not meet you on 12th. Today is 8th. Oh, no, maybe I do not meet you on 15th, then I will meet you on 22nd. Okay. Thank you. And good
Yes,
night.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Good night, sir.
Good night. Bye.